Europa, a moon of Jupiter, and Enceladus, a moon of Saturn, are fascinating celestial bodies. Both moons have evidence of vast oceans beneath their icy crusts. These subsurface oceans are heated by tidal forces from their host planets and neighboring moons, creating a potential environment for life. The icy surfaces of these moons are bombarded by radiation, making them seemingly inhospitable, but the oceans below could tell a different story. Despite the harsh radiation on Europa and Enceladus, life signs in the form of organic molecules might survive just beneath the surface ice. NASA's recent experiment suggests that if these oceans support life, signatures like amino acids and nucleic acids could endure the ionizing radiation. This discovery is crucial because it means that robotic landers sent to these moons wouldn't need to dig very deep to find these life signs, making the search for extraterrestrial life more feasible. NASA's experiment involved mixing amino acids with ice chilled to about -321 degrees Fahrenheit, or -196 degrees Celsius, in sealed airless vials. These samples were then bombarded with gamma rays, a type of high-energy light at various doses. The team also tested the survival of amino acids in dead bacteria and in ice mixed with silicate dust. These experiments provided pivotal data on the rates at which amino acids break down, known as radiolysis constants, under conditions similar to those on Europa and Enceladus. Based on our experiments, the safe sampling depth for amino acids on Europa is almost 8 inches, or around 20 centimeters, at high latitudes of the trailing hemisphere, said Alexander Pavlov of NASA's Goddard Space Flight Center. For Enceladus, subsurface sampling isn't required as amino acids can survive radiolysis at any location less than 1/10 of an inch, or a few millimeters from the surface. This means that future missions to these moons could potentially find life signs without extensive drilling. Amino acids are essential to life as they are used to build proteins, which in turn make enzymes that regulate chemical reactions and structures. These molecules can be created by both biological and non-biological processes. However, finding certain kinds of amino acids on Europa or Enceladus would be a strong indicator of life, because they are used by terrestrial life to build proteins. The presence of these amino acids could suggest that the subsurface oceans of these moons harbor life. The experiments revealed that amino acids degrade faster when mixed with dust, but slower when coming from microorganisms. This suggests that bacterial cellular material might protect amino acids from the reactive compounds produced by radiation. Slow rates of amino acid destruction in biological samples under Europa and Enceladus-like surface conditions bolster the case for future life detection measurements by Europa and Enceladus lander missions, said Pavlov. The team also found that the rates of potential organic biomolecules degradation in silica-rich regions are higher than in pure ice, indicating that future missions should be cautious in sampling these areas. The findings from NASA's experiment are promising for future missions to Europa and Enceladus. The data suggests that life signs could be found just beneath the surface ice, making the search for extraterrestrial life more accessible. Future lander missions should consider the varying rates of amino acid degradation in different regions and prioritize areas with pure ice over silica-rich locations. With these insights, the quest to find life beyond Earth takes a significant step forward, bringing us closer to answering the age-old question: are we alone in the universe?